So in this last video, we're going to be talking about enzymes. Enzymes are needed to speed up chemical reactions inside cells. Chemical reactions in a test tube may take minutes or hours, and this simply isn't fast enough for cells. And so most biological chemical reactions can only occur fast enough in the presence of an enzyme. Enzymes are protein catalysts, and what this means is that they bring reactants together in precise orientations. And your textbooks call these reactants substrates, so I use these words interchangeably. But when substrates are in these precise orientation, this makes the reaction more likely to occur. Enzymes are also specific for a single kind of reaction. I'm going to start by telling you about my favorite enzyme. This is myeloperoxidase, or MPO, and it's a peroxidase enzyme. It's made up of 4 polypeptides, and so what you're looking at here is its quaternary structure. MPO is expressed specifically in a cell called a neutrophil, which is a type of white blood cell. And one of the reasons it's interesting is because it contributes to the green color of pus. So this enzyme binds to a small molecule called heme. And when it binds, it actually forms a covalent bond with heme, and this gives the heme unique spectral properties that turn it green and turn the molecule green. And so what happens is during an infection, you get neutrophils arriving at the site of infection. They're fighting the pathogen, but a lot of them die. And when they die, they burst open and release their MPO. So if you have a lot of dead neutrophils, you have green pus. It also catalyzes the following chemical reaction, which I think is a very cool chemical reaction. What it does is it takes hydrogen peroxide and a chloride ion and turns it into HOCl and water. And HOCl, or hypochloric acid, is actually the active component in bleach. So this is a really powerful antimicrobial that's made inside your cells that's capable of killing pathogens. So this is why I think MPO is cool, but we're going to talk more about enzymes and all of their properties next. So for an reaction to take place, reactants or substrates need to collide in a precise orientation, and they need to have enough kinetic energy to overcome the repulsion between electrons in atoms that come into contact as they form bonds between one another. And enzymes help reactions clear these 2 hurdles. Enzyme catalysis has 3 steps. The first step is called initiation, and that's shown here. The substrates are precisely oriented so that they can bind into the active site. Step 2 is called transition state facilitation. And this is when there are interactions established between the substrate and the R groups extending into the active site of the enzyme. And this lowers the activation energy needed for the reaction to take place. The final step is called termination, in which the products are formed, and released, and the enzyme can be reused. And we're going to talk about these 3 steps in more detail. So during initiation, what happens is that enzymes bring together specific substrates. And so here's an example of glucokinase. And what this enzyme does is it binds glucose and ATP, and it binds them in their active site. And this enzyme is going to help these 2 molecules come together in a precise orientation that's going to allow them to break some bonds and reform others to make a product. When these substrates bind to the enzyme, the enzyme undergoes a conformational change. And when the substrates are bound to the active site, it goes from this open form to this clamped form around the substrates. And this change in shape, or change in conformation, is called the induced fit model. The induced fit model is favored over an older model called the lock and key model because it accounts for an enzyme's dynamic and flexible properties, right? These shape changes that take place when an enzyme has bound to its substrate. So the next step is called the transition state facilitation step. And in this step, substrates bind to the R groups in the active site via hydrogen bonding and other interactions. And this forms, so here's our substrates. This forms an intermediate condition called the transition state. And so A forms kind of a temporary bond with both B and C. Activation energy is a kind of energy that's needed to strain the substrate's bonds so they can reach this transition state. And enzymes lower the activation energy. This is a really critical point. The free energy of the system, or whether or not the reaction is spontaneous, or the change in energy between the substrates and the products, it never changes. And this might be a quiz question. Once the products are formed in termination, the products are released, but the enzyme is never consumed, and so it can keep processing new substrates. So again, this is our free energy equation we talked about before. And so we have our reactants that reach this transition state, and there's a certain amount of activation energy that's required to strain those bonds to reach the transition state. And then we have our products. And in this equation, or in this free energy graph here, the reactants have higher potential energy than the products, and so this reaction would occur spontaneously. Enzymes would change the shape of this graph, and the part of the graph they would change is actually the activation energy. So this is a really critical point. The activation energy is lower in the presence of an enzyme, but the delta G, whether or not a reaction is spontaneous or not, does not change. And so that part of the graph remains the same. So what are the limits for enzyme activity? The speed of an enzyme catalyzed reaction looks something like the graph below. So here in blue, first I'm going to show you an uncatalyzed reaction. So here we have substrates in a test tube without the enzyme. And you can see that the product will form, right? And it's somewhat dependent on the substrate concentration, but the product actually forms pretty slowly, right? Here's an enzyme catalyzed reaction shown in red, so this upper line here. This increases linearly at low substrate concentrations. So this reaction is dependent on the concentration of the substrate. But the reaction or the enzyme activity actually slows as the concentration of the substrate increases and reaches maximum speed at these high substrate concentrations. So all enzymes show this type of, we call it saturation kinetics. And this is because active sites cannot accept substrates any faster. No matter how large the concentration of substrates gets at this point, there's simply not enough enzymes to make more products. And reaction rates level off because all of the available enzyme molecules are being used. The enzyme can also be limited if its shape changes. Recall with proteins that the structure is critical to its function. And the structure is mediated by bonds that help the protein fold. And these bonds can be interrupted by changes in temperature and changes in pH. So temperature affects the folding of a molecule, so it can change or it can decrease the enzyme activity, but it can also affect the movement, the kinetic energy of enzymes and substrates, and increase their activity, or the enzyme's activity. Similarly, pH can affect the enzyme's shape and reactivity. So pH can change the status of those ionic and basic R groups and disrupt their interactions with each other, break apart ionic bonds, or maybe break apart other bonds. And so each enzyme has evolved to be best in its surroundings. And so each enzyme has an optimal temperature and pH. And let's take a look at that. So here's two graphs. Okay, one's looking at enzyme activity or of a specific chitinase and its dependence on temperature. And the other one is looking at its dependence on pH. Okay, so this purple line here, This shows the enzyme activity of chitinase from a bacteria that lives in a cool, neutral environment. Okay, so this enzyme works best at around 45 degrees Celsius. And then it doesn't work so well when the enzyme is brought into a higher temperature. Okay, this is compared to if you isolated this enzyme from a bacteria that lives in a hot and acidic environment. Well, you see this enzyme, it actually peaks with its activity at 60 degrees. So it functions even better 10 degrees at a 10 degree higher temperature. But this also falls off, and that's because at these higher temperatures, the proteins are going to be natured. So they're going to be broken down into their primary structure and no longer functional. We can look at the same thing with pH. We'll go through this slowly, but this the enzyme from bacteria that lives in a hot acidic environment works best at a pH of 2, and the enzyme isolated from bacteria that lives in this cool neutral environment works best at a pH slightly above 6 or maybe 7. And so you can see they're adapted to their environments, but under extreme conditions, neither one works very well. So in addition to temperature and pH, enzymes can be regulated by regulatory molecules, and these can control when and where an enzyme functions. As an example, let's talk about the phosphorylation of enzymes by using ATP as cellular currency. So in this example here, we have an enzyme, and it's shown in gray, and we're zooming in on the active site. And so these balls here represent amino acids, and the rest of the amino acids are represented by this orange squiggly line. And right now, this enzyme is off because this activation loop blocks this active site here. But when the molecule becomes phosphorylated by ATP, the activation loop moves, and now the substrates can bind to the active site. Phosphorylation may activate an enzyme, just like in the example we talked about, or it may work in the reverse and change the conformation so that the enzyme can't bind to its substrate, in which case it inactivates the enzyme. We'll talk more about enzyme regulation when we talk about metabolism, but for now, we're going to focus on how enzymes are influenced by concentration of a reactant, as well as their own concentration, temperature, and pH. And we'll talk more about this in lecture as well as in the lab. And if you're interested, you can read ahead in your textbook to learn about how cofactors, coenzymes, prosthetic groups, and different types of inhibitors work to regulate the function of enzymes. So that's it for this video, and I look forward to seeing everyone in class. Thanks, bye.